Hey biology kids, good luck on your test tomorrow. Let's go through the review sheet. So first thing is defining some key terms. Evolution is defined as a change in alleles or genes over time. And by time we're talking about many, many generations, not just in the next 5 minutes. Homologous structures are common features, and those are due to a common ancestor. Vestigial structures, these are structures that are remnants of a common ancestor but they no longer serve their function. We'll get to some examples of that later. Last, we've got analogous structures. Those are common features due to the environment, not a common ancestor. Then we've got the Hardy-Weinberg principle, and that is simply math, and it's a way to say that the population will not evolve if all of the conditions are met. And we've got genetic drift, which is simply the random change in allele frequencies. Founder effect is simply when we have a small subpopulation that leaves the original population. Maybe it goes to colonize a new island or anything like that, and it does not have the same gene balance or allele frequencies as the original. Selection. It's based on a couple of key observations. First one is variation. He noticed that there were differences among members of a species population. Brown hair, blonde hair, so on and so forth. Next thing was heritability. He said that all of those differences are due to genetics. They had to be inherited from your parents. It's not something that you could just do on your own during your lifetime. You can't force your hair to start growing out brown or force your hair to start growing out blonde just because it's convenient. Then we've got overproduction. More offspring are produced than will survive. Next one is reproductive advantage. And that simply says that if you survive, you have some sort of advantage that you can pass on. Competition, if there's ever a limited resource, which is like always in nature, he said the fittest will survive. Next, comparative anatomy is one piece of evidence for evolution. Comparative embryology and comparative biochemistry. With embryology, what we learn is that embryos of mammals, fish, reptiles, etc., we have similar developmental stages. That tells us that we share a common blueprint. We have the same instructions for how to develop early on, and then we branch out. And then with biochemistry, that's basically looking at DNA. And we're looking at mutations. If we have similar mutations, different mutations, we're looking for how much of our DNA is similar and things like that. Then we're going to take a look at adaptations and some examples of that adaptation. So camouflage, that's pretty obvious for us. We live in Minnesota, we hunt. Mimicry we're going to see in frogs, and then antimicrobial resistance is the same as antibiotic resistance. It says that we've got 6,000 fish total, 2160 are homozygous recessive, and they have diamond-shaped scales. Calculate the following. Well, homozygous recessive, if I look at my equation, represents q squared. So I know that this is going to be 2,160 out of my 6,000 fish. When you break that down, put it as a fraction into your calculator and simplify it, that gives you 0.36. So 36% of this population has the weird recessive scales. Now I'm going to go over here. To find q squared to q, you have to take the square root of 0.36, which gives you 0.6. I know that p plus q has to equal 1, so 1 minus 0.6 gives me 0.4. So to find p squared, here's p. I simply have to take 0.4 squared, which is 0.16. And then 2pq, 2 times 0.4 times 0.6 equals 0.48. List a few factors that influence or control genetic drift. Genetic drift is controlled by population size. It's also controlled by allele frequencies. And migration has a huge part of it. Migration, natural disasters, anything that's going to limit the number of people in our population or change the balance. Explain or give an example of the founder effect. A great example of founder effect would be in the Quaker population that had polydactyly, extra fingers. So what happened there is that the small subpopulation moved out, and when they did that, they took a very weird balance of alleles with them, and that changed the whole population. All right, our first selective pressure is stabilizing selection. If something is stable, that means it's going to stay pretty much the same. So in that case, let's say we've got a bell curve of fish of all different types of sizes, and the little ones are going to die off, and the big ones are going to die off, but everybody who's average stays the same. So in here, it is a benefit to be average. And what happens over time is the population becomes more average. Then we've got directional selection. Directional selection, if we've got a bell curve like this, only one extreme or only one variety has an advantage. These guys all die off, these guys all live. So the benefit is to be one extreme. So our example in class was anteaters, and so the population would shift to all have long tongues in anteaters. Then we've got disruptive selection, where here's our bell curve, and being this way is good, being this way is good, but anything that's average or down the middle, or half and half, is very bad. These guys are all going to die off. So both extremes have a benefit. And this is going to actually split our population. Then we've got sexual and artificial selection. Sexual selection is where fitness is determined by a choosy female. And that can lead to something called runaway sexual selection, which is pretty bad stuff. Which is where the male appearance is more important than his survival. So that would be like a male peacock whose feathers get so ridiculously large that he can't even fly away from predators, but the girls like it, so it continues to happen in the population. Then we've got artificial selection, which is also known as selective breeding. This is human-influenced evolution. This is where we choose who lives and dies. This is where we choose who breeds and doesn't breed. So some examples of that would be with dog breeds. Could also be with crops or plants, like we mentioned in class with the wild mustard plant becoming broccoli and Brussels sprouts and everything else like that. All right, next up, prezygotic and postzygotic isolation. Pre simply means before, and post means after. Zygotic simply refers to the fact of fertilizing an egg. So prezygotic means before fertilization. So an example of prezygotic isolation would be like two birds with different mating seasons. And then post, an example would be, all right, a horse plus a donkey can come together and make a mule, even though that mule is not actually successful as its own organism. They do have an offspring created. It's just sterile and can't reproduce on its own. Then we've got allopatric and sympatric speciation. Allopatric means different homeland, and sympatric simply means same homeland. So the way we think that happens is this one is caused by geographic isolation. They just simply can't reach each other anymore. So they can't mix, they become gradually more and more different over time, and that's how we think that causes different species. A good example of that is the squirrels in the Grand Canyon. And then sympatric speciation, we think this is mostly due to reproductive isolation. They're in the same place, they're interacting all the time, but we see, just like above, either they don't recognize each other or they can come together, but it doesn't make successful offspring, and that results in two different species. What is the difference between coevolution and convergent evolution? Here's a tip. Think about like cooperating. They're working together. And then convergent, think about the word converge, which means come together. I ran out of room. Okay, so coevolution. Good example of that would be like a acorn, which comes from an oak tree, and a squirrel. That's the example I gave in class. Or you could do a hummingbird and a flower. Tons and tons of examples. But either way, we're going to have two species, SPP is species, that affect the evolution of each other. So what would happen with the acorn and the squirrel is that the acorn is bitter, so the squirrels that get all the acorns and survive better are those that don't taste it. But the acorns don't want to be eaten, so the bitterest ones are the ones that are left on the floor. They go back and forth. Bitter, no taste buds. More bitter, less taste buds. More bitter, less taste buds. And so they're affecting each other's evolution. And then we've got convergent evolution, which simply means that they're going to come together from very different places. So this would be like a dolphin and a shark, or you could also say like a bat and a bug. They both have, oops, you can't see that. Dolphin and a shark both have fins. A bat and a bug both have wings, but they're not closely related. So two unrelated species, or at least not closely related species, develop the same structures because they live in the same environment. Bi meaning two, pedal meaning on your feet, so bipedal, walking on two legs. Oh my gosh, guys, seriously. Okay, we diverged 5 to 8 million years ago. 5 to 8 million years ago. That's the last time we had a common ancestor with modern apes. We are all primates, us, apes, chimpanzees, etc. But we're also all mammals, just like dogs and cats and pigs and cows and everything else are mammals. So we have that same common ancestor, but we have been separate from them for 5 to 8 million years.